The overall goal of this procedure is to provide a convenient way to control ion channels expression in heterologous systems while avoiding lengthy cell culture complications, thus providing a way to control conditions between experiments and generate more replicable results.
This method can help facilitate research in the ion channel field, such as characterizing channel properties in the pharmacology, since the current methods are either complex or show inconsistent protein expression.
The main advantage of this technique is that we can achieve controlled expression levels of a wide variety of proteins tailored for individual experiments' goals within a relatively short time.
To begin this procedure, transfer T-Rex 293 cells to a 12-well plate prepared with 0.9 milliliters of full DMEM per well for generating 50% confluency, and incubate the cells overnight at 37 degrees Celsius in a CO2 incubator. Next, prepare a transfection mixture composed of DMEM, the gene of interest, shown here TRPV1, an inert plasmid, and the lipid transfection reagent. For electrophysiological experiments, include EGFP in mammalian expression plasmid in the transfection cocktail in order to visualize the cells that successfully undergo transfection. Incubate the transfection mixture at room temperature for 30 minutes. Then, transfect the cells by pipetting the transfection mixture dropwise onto the plated cells in the 12-well plate. Rock the plate vigorously to ensure a homogeneous dispersion of DNA and transfection reagent. Afterward, incubate the transfected cells overnight at 37 degrees Celsius. The next morning, if performing